Welcome back. So we're in the middle of a series of vignettes, all of which share the objective of trying to examine the various hypotheses that we were able to come up with for what might limit the rate of bacterial cell division. So what we're going to do right now is we're going to examine the question of RNA synthesis, and we're going to do that in several pieces. I want to think about RNA synthesis both in the context of messenger RNA, shown over here, but also in the context of ribosomal RNA synthesis. So my claim is that both of those put demands on the current budget of RNA polymerases, and the question is, are there enough RNA polymerases around to keep up with these processes in order to fuel the cell division process? So what I want to do is let me start by thinking about transcription of mRNA. So let's see, how do we want to think about this? So the thing that I want to say is that there are on the order of between 2,000 and 10,000 mRNAs. In cells, so this is slow growth, and this one is fast growth. And another point that needs to be made is that mRNAs have a relatively short lifetime in bacteria. So the lifetime of a bacterial mRNA is something like 200 seconds, I'm going to say. And so what this means is that in order to maintain a steady state of mRNAs, the RNA polymerases are constantly working at generating new copies of the mRNAs of interest. And so I want to follow up on our dynamical systems worldview. So we're going to make an aside, and we're going to think about a dynamical systems perspective on mRNA dynamics. So in a nutshell, what I would say is that the number of mRNAs that we find in the cell now, so this is number of mRNAs now, or at time t plus delta t, is equal to the number of mRNAs at time t, and then plus the number of mRNAs that are synthesized during the time delta t, and then minus the number of mRNAs that are degraded in time delta t. So I'll put a green mark under that one, and I'll put a red mark under this one to signify growth or transcription and decay. Now we can think about this in the language of dynamical systems that I already introduced earlier by making a plot of the rates as a function of the current number of mRNAs. So for example, what we have is that the growth rate is basically constant. In other words, R, this is the R process, it doesn't care how many mRNAs are already present. The rate at which new mRNAs are being produced is R. And on the other hand, the decay is a linear function of M. In other words, the decay, the more mRNAs I have, the more that will decay in every time. So what we learn from this is the existence of a fixed point, which I'm going to refer to as M star, and that's the point at which the number of mRNAs is in steady state, where growth is balanced with decay. And we can actually solve for this. Writing our differential equation, we have that dm by dt is equal to r minus gamma m. And in steady state, what does steady state mean? It means that it's not growing. So dm by dt is equal to 0. So that implies that r minus gamma m star is equal to 0, or that m star is equal to r over gamma. Now, why did I go through this? I went through this because now we're in a position to actually estimate the parameter r, meaning the number of mRNAs that are produced per minute or per second to maintain the overall steady state population size. So I know that m star, let's say that that's 10 to the 4th, and I know that gamma is 1/200th. Well, let me actually use gamma is 1/3 per minute. So I'm basically saying that the lifetime of the mRNAs is 3 minutes. So then that means gamma, the decay constant, is 1 over the lifetime, so 1/3 per minute. So this allows me then to find r as m star times gamma, which is equal to 10 to the 4th divided by 3. And that's in units of mRNAs per minute. So something like f times 10 to the 3 mRNAs per minute. So that's the steady state rate at which mRNAs need to be synthesized by the RNA polymerases. Now let's think about that a little bit. A typical gene, is, let me see, something around 1,000 nucleotides. The reason I say that is that I know that a protein, so typical protein, is 300 amino acids. And so given that, we know that there are 3 nucleotides for each amino acid, so 3 times 300 is 1,000. So there's 1,000 nucleotides, and so that's the length of a typical gene. And that and RNAP synthesis rate is of order 60 nucleotides per second. And what this tells me is that it's roughly 20 seconds to synthesize an mRNA. Okay, so why do I say that? Because if it's going 60 nucleotides per second, then that means it'll travel 1,000 nucleotides in roughly 20 seconds. 20 times 60 is 1,200, so it's a little bit less than that. So what that means is that the residence time of RNAPs on mRNAs, they're taken out of circulation for roughly 20 seconds. So my final little statement here is to say that we can now figure out what the standing pool of RNAPs that must be used in order to keep the busy process of transcription going. So I want to just make a first go at telling you about something called Little's theorem. So Little's theorem is a really cool idea that relates flux to population size and residence time. And the easiest example I know of to illustrate this is to think about Caltech undergrads. So the flux is 200 undergrads per year, and we'll leave the population of Caltech undetermined for the moment, and the residence time is 4 years. And so that tells me that N, the population size, is 4 years times 200 students per year, which is the flux. So that's 800 students, something like that. So you might be saying, well, what does that have to do with this RNAP problem? But here, I know that the flux is, I need 3,000 transcripts per minute. So that's the flux. And that should be equal to the population size. And then 1/3 of a minute is the residence time, because I said it was 20 seconds. And so this means that basically I need 1,000 RNAP. That's the population size that would be needed to keep up with the process of transcription of mRNAs. Okay, so let's just take a quick look at this. So here's the proteomic data on the RNA polymerase subunits. And what you can see here, for example, if you look at the beta or the beta prime subunits, that there's a few thousand copies. So what's my point? I want to keep my eye on the prize. The point is that in order to keep the pool of mRNAs between 2,000 and 10,000 mRNAs current, we need on the order of 1,000 RNA polymerases. So that's already well in hand. The second question is a little bit more subtle, and now we're not talking about mRNA, we're talking about ribosomal RNA. Now we need to remember they're on the order of between, say, 7,000 and 70,000 ribosomes in cells, 7,000 for slowly dividing cells and 70,000 for the fastest dividing bacterial cells. The ribosome is a little bit over 2 megadaltons, and 2/3 of that is RNA, ribosomal RNA. And that ribosomal RNA, as you can see at the top here, that ribosomal RNA is comprised of one very long RNA, which is 2,900 nucleotides long. And what we're going to do here is we're just going to try to reason out whether or not the RNA polymerases can keep up with the business of making, for example, 20,000 new ribosomes in the time allotted by the cell cycle. So the first little calculation shown here is just to say if I only had one RNA polymerase and it had to do the 2,904 nucleotides, it would take 48 seconds to travel that distance. And so if I only had one of them and I needed to make 20,000, then I need to take 20,000 times 48 seconds. And as you can see, that's 15 days. Okay, fine. So it would take a long time. Now let's imagine alternatively that we were loading the ribosomal RNA gene as frequently as we possibly could. Now, what do I mean by that? The RNA polymerase is about, let's say, 50 or 60 nucleotides wide. It's moving at 60 nucleotides per second. So that means that the fastest we could continue to load RNA polymerases onto a ribosomal RNA gene is about 1 per second. So we're making one new ribosomal RNA every second, but we need 20,000 of them, so 20,000 seconds would be required, which is roughly 5 hours. So again, that's too long, but of course that's presuming that there's only one copy of the RNA, the ribosomal RNA gene. Just as an aside note, part D here, that's just to give you a sense of the transcription off of these genes. But the real resolution of the puzzle comes in the form of the very interesting point, which is that there's not only one copy of the ribosomal RNA genes. There's actually 7 of them in E. coli. So as you can see in this calculation, if I have 7 copies of the ribosomal RNA gene, each of which is being loaded at a rate of 1 per second, then we've now brought the time of synthesis of the entire pool of ribosomal RNAs down to 0.8 hours. But it's even better than that. And we're not going to go into this in detail, but it's part of the reading that comes along with this particular series of vignettes, which is that these ribosomal RNA genes are close to the origin of replication. What that means is that there's actually there's more than 7 copies present because of the nascent synthesis of further, what I'm saying is new replications have already occurred before the cell divides. And so you might have as many as 8 copies of each one of these ribosomal RNA gene clusters. In that case, the total time ends up being 0.1 hour. So what's the point of all this? The point of all this, so what we've done in this little vignette is basically to address the question of the transcription hypothesis, both for mRNA. We found that only 1,000 RNAPs are required for that. We examined ribosomal RNA synthesis, and my assertion is that the RNAP pool, which we looked at here, is sufficiently large to keep up with the demands of transcription. And note that with increasing growth rate, as you can see here, the trend is that the number of RNA polymerases goes up with increasing growth rate. So parallelization comes to the rescue again. So the final hypothesis that we're going to examine is the protein synthesis for ribosomes. In other words, a ribosome has to make a new ribosome, and that's going to be the target of our final hypothesis that we'll think about.